Hello everyone, welcome back to another class on delta notch signaling during regeneration. So we have mentioned about delta notch signaling multiple times in the previous classes because it is one of the most important signaling events that governs the regeneration as well as embryonic development, and in different tissues it plays different roles. Say for example, in some tissues it is important to retain the homeostasis. That means the day-to-day affair of the tissue has to be taken care by delta notch signaling, whereas in other cases they are important for transforming the stem cells into a differentiated cell types. They usually, their expression is somewhat referred to as salt and pepper way of induction. Means you take a black pepper powder and you sprinkle some salt, what pattern you get, it something like that is their expression. So let us quickly visit the delta notch signaling pathway in an overview manner. The picture seems a little complex, but don't worry, we can lighten it out. It's a schematic diagram of notch signaling pathway following the ligand receptor translocation, that is of the NICD domain. And there are different proteins comes into picture. ADAM stands for a disintegrin and metalloproteases. ANK is the one which is anchoring repeats. COR, which means co-repressor. CSL means a bunch of genes, CBF1, SU, LAG, DSL means delta, serrate, LAG2, and MAML, MAML means mastermind, like NICD means notch intracellular domain, NLS means nuclear localization signal, NRR means negative regulatory region, and PEST stands for purine glutamic acid serine threonine. These are all well accepted terminology, and TMD stands for transmembrane domain, etc. So you can see here there is a EGF like repeat bearing delta molecules, that is, this is one cell and it is present, and this is another cell that has also have got EGF like repeats, but it is called a notch. So the delta must interact with the notch for any activity to happen, and there is also a step in the maturation of the notch ligand, and for which the ADAM family enzymes comes into picture. And what we should understand, because of the deltas binding onto the notch, you end up getting a cleavage or the cutting of this notch intracellular domain, which is favored by gamma secretase. It's a S2, S3 and S4. That means different sites of cleavage that happens, but the final action is done by gamma secretase, and that triggers the release of the notch intracellular domain. That is, this portion will be released. This portion will be released, and that will go into the nucleus NICD, and that can turn on bunch of genes. That is, it can be Hess, Her, many genes are there, and this is what the typical way of functioning of NICD or the notch signaling events. So the diagrams that is showing the how delta notch signaling contributes to endocardium, that is the signaling events that is happening after a myocardial infarction. If there is a ischemic heart injury, the blood supply to the heart muscles are stopped, how endocardium or myocardium comes into picture and contributes to retaining the homeostasis, and different injury and stress models. So we can see one situation is A, that is the after ventricle ablation, and the B is after ventricle cryo injury. So what we are seeing here is notch activation results in the non-cell autonomous initiation of myocardial ERBB2. Remember, we saw in the previous class about the ERBB2, which is whose ligand is not known, but it is important in the EGF signaling, ERBB2 and the BMP signaling, which is responsible for the cardiomyocyte reprogramming and proliferation. So as you can see here, and then upon insult in the panel B, notch signaling is significantly activated in the endocardial cells. So endocardium is the wall of the heart muscle which is facing the blood, that is the lumen of the heart. And then the myocardium, then comes the epicardium. And above the epicardium, the layer is pericardium. So endocardial cells which restricts the expression of inflammatory factors and macrophage recruitment and coordinates with a gene or a protein called SERPIN1 to control endocardium maturation and cardiomyocyte proliferation. This is its normal role. So what we can see in this picture is this is the cardiac lumen, and there is lot of cells that is exposed to this cardiac lumen, and the notch is one of them. And the notch, when it gets activated, you end up getting the NICD, which in turn will be facilitated. More and more of notch will be produced into the cell, and various signaling events can trigger an anti or a negative role onto the migration of NICD to the nucleus. That is possible, but in general this can trigger an ERBB2 mediated NRG1 and the ERBB2. This we are talking about in a regenerating model. NRG1 can bind onto ERBB2 in the case of regenerative animals such as zebrafish, that you should keep in mind. And the BMP signaling that triggers the dedifferentiation and transdifferentiation of the myocardial cells, MCs, myocardial or the cardiomyocyte cells, it happens. And what happens if you have given a cryo injury, the SERPINs molecules, they comes into picture for the EC maturation, EC proliferation, and also in the neighboring cell, what happens, it can also inhibit the myocardial cell proliferation. The progenitors have to be formed for the regeneration to happen, but the epicardium and myocardium we are talking about. In epicardium cell, endocardium cell and also the myocardium, they interact with each other. They are the one which is triggering this delta notch signaling for their intercellular communication. Notch triggers an NICD production, and it also helps in the release of various factors, and it can negatively influence the inflammatory factors and macrophages. Like we saw in the case of hippoap signaling, in the heart you need to have negatively influence. Too much of inflammatory response is not welcome, hence they have to block the inflammatory factors, and that is also favored by the delta notch signaling. And this is the diagrams that showing the functions of endocardial notch signaling and various heart and homeostasis as well as stress injury models. So this panel C is after ventricle amputation. So the inhibition of the endocardial notch signaling could cause reduced expression of Wnt antagonist. Wnt signaling now we are talking about. What are Wnt antagonists? They are WIF1 and NOTAM1B. NOTAM, you may remember, we have studied in the invertebrate regeneration also. Thus, the Wnt activity was increased and could inhibit cardiomyocyte proliferation and heart regeneration. This regenerative defect could be partially rescued after treatment with Wnt inhibitor IWR. So in the panel D, what you are seeing is the notch signaling could control the balance between fibrotic and regenerative repair in the adult heart. Interestingly, what we should note is that the Notch1 knockout, if you knock out the Notch1 in cardiomyocyte, it does not affect the Jagged1 induced anti-hypertrophic and anti-fibrotic responses. They happen because naturally various other signaling, like we have seen it, YAP signaling comes into picture, they can also contribute. The cell types responsible for the beneficial functions of the Notch1 overexpression yet to be identified, and the endocardium stands out a good candidate for this proposal. So you can see here the notch signaling, once kick-started NACD, they can release the Wnt inhibitors so that in the myocardium, the Wnt signaling will not act very effectively because the Wnt signaling is prevented, hence they will not proliferate. And you can also see in the case of after jagged overexpression, if you do it, that is you are forcefully allowing the delta notch signaling to go up in the endocardial cells, then what you end up getting is that there will be lot of influence that is occurring in the myocardial precursor expansion will be favored, and also the myocardial proliferation will be favored, and you also end up getting lot of fibrosis, that is the fibroblast cell and the fibrosis that is coming into the scenario. So in one signaling can do in either way. Minimum it does one job, maximum it does another job. So this is what you should keep in mind, that the same signaling will be able to perform different tasks. So the role of notch signaling pathway in recovery of cardiac function after myocardial infarction. So myocardial infarction, we all know, it is the disability of the heart muscles to perform. It is normally occurring because of a lack of blood supply, that is typically known as the ischemic heart injury, and it's a pathological process evidencing as a massive death of cardiomyocytes associated with hypoxic and oxidative stress. The formation of areas of fibrosis that leads to heart failure. We know that there are some mechanisms that contribute to the functional repair of the heart, and some mechanisms may not restore the myocardium, but it can prevent the onset of too much of inflammation. In most mammals, including humans, the notch signaling pathway has a cardioprotective effect. It is involved in the formation of heart during embryogenesis and in the restoration of cardiac function in the myocardial infarction due to some steps. That is step number 1, reducing the oxidative stress. Step number 2, prevention of apoptosis. And step number 3, regulation of inflammation. And step number 4, containment of fibrosis and hypertrophy of the cardiomyocytes. And also the step number 5, the tissue revascularization. And 6, the regulation of proliferation and differentiation of the cardiomyocytes. In addition to this, the notch signaling pathway interacts with various other signaling events, like we saw how it is influencing the Wnt signaling by producing Wnt antagonists, and it can also get involved in several pathogenic events of myocardial infarction and subsequent cardiac repair. So even in mammals, even though it cannot regenerate, it has a cardioprotective role. And if you look into the tissue and cellular events of myocardial infarction, it has multiple phases: ischemia, hypoxia, inflammation, termination. This is the broad phases. After ischemic exposure, an increase in the level of ROS and the lack of ATP that lead to cell death and release inflammatory mediators. During the inflammation stage, the following processes occur. Let us see what are they. Phagocytosis of the dead cells, that must happen, and the debris also should be cleared, and also it should form new blood capillaries so that new cells can survive. At the termination stage, some conditions restoring the integrity of the myocardium by replacing the dead cardiomyocyte cells, CMCs, with fibroblast and hypertrophy of the surviving CMCs. Hypertrophy means expansion of the size so that the damaged portion can be restored back. And this is a schematic representation. We have already seen it. This is a quick revisit of the delta notch signaling, because delta notch signaling, depending upon species, it can have delta ligand, delta-like ligand DLL, and JAGED, JAG. All these things can come into picture. And eventually the ligand, no matter what it is, the notch has to get activated or bound by this ligand, and the notch receptor can be notch 1 to 4. The notch intracellular domain is cleaved and binding when the notch is bound with its ligand, and the activated NICD enters the nucleus, where it interacts with the intracellular regulatory complexes, after which the transcription of the notch target genes takes place. As you can see here, JAGED is there, DLL is there. They are all acting onto the notch, and the ADAM and the gamma secretase ADAM cleaves here and the gamma secretase cleaves here, and this NICD is freed. So ADAM is the first cleavage, gamma secretase is the second cleavage. And if NICD goes into the nucleus, it can work in canonical way, and also it can do several non-canonical pathway also. many genes are turned on, HES, He, NKX, etc. And lot of this cleavage of the notch, when it is maturation, there is multiple cleavage phases that takes place. That is furin cleavage and Adam cleavage and gamma secretase cleavage, etc. So that's how a notch gets matured. See, the role of notch signaling pathway in reducing oxidative stress at the cardiomyocyte cells. So we can see when notch is turned on, it can favor NACD and LKB1, and it can trigger an AMP kinase and also ATP production. And it can also negatively affect TNF alpha, which is a pro-inflammatory cytokine, and which in turn inhibits MFN2. So technically notch is activating MNF2, which is helping in the mitophagy. And notch can also activate AKT, which is production of several antioxidant ferments. So launching notch signaling pathway enhances the synthesis of MFN2 protein responsible for mitophagy. The Notch1 receptor can bind to the tumor necrosis factor alpha and block its action. And the production of antioxidant enzymes is controlled through the AKT cascade, and the energy conservation in the cells is achieved by activating MAP kinase cascade. So this much roles the notch signaling can trigger. So if you look into the significance of the notch signaling pathway in preventing cardiomyocytes apoptosis, through the transmission of the notch signal, a decrease in the expression of pro-apoptotic BAX and an increase in the expression of anti-apoptotic BCL2. Pro-apoptotic BAX is decreased and anti-apoptotic BCL2 is increased, is achieved. Additionally, the prevention of apoptosis can be achieved by increasing the expression of genes associated with autophagy. Inhibition of the pro-apoptotic tumor necrosis factor alpha, which is also a pro-inflammatory molecule, also helped to prevent the apoptosis. Like you can see here, notch activates Becklin, LCM-I, LCM 2, and also it can trigger the autophagy, and also it upregulates BCL2, which is anti-apoptotic, and downregulates BAX, and so that it can safeguard the cells from apoptosis, and it blocks TNF alpha, which is a facilitator of apoptosis. So if you block it, the apoptosis will be prevented. So these are all the roles played by the notch signaling on apoptosis, how to influence the apoptosis. Now if you look further, notch pathway have a significant role in regulating the inflammatory response. Like in the previous class, we have discussed a lot about M1 and M2 macrophages. We have discussed M1 macrophages are more bringing in pro-inflammatory environment, whereas M2 is bringing in more of anti-inflammatory environment. So let us see how it is contributing. So the notch signaling promotes the acquisition of a pro-inflammatory M1 macrophage phenotype that secretes inflammatory mediators. So if you have active notch signaling, what happens is the production of M1 macrophage takes place, which can secrete IL-6, MCP1, TNF alpha, so many other pro-inflammatory cytokine. And if there is DAM signaling that is triggered, then you end up getting more of the M2 macrophages. M2 macrophages, if it is present, they are anti-inflammatory. But remember, for regeneration to happen, you need to have pro-inflammatory events triggered soon after injury. And this comes later. IL-10, IL-RA, these are all derived from the M2 macrophages. So the notch signaling pathway very selectively influence the upbringing of M1 macrophages. And we can also see the myocardial remodeling in the fibroblast, also the mechanism of inhibition of myocardial remodeling in fibroblast. We know fibroblast bring in the fibrosis, and fibroblast bringing in the scar formation, etc. after a myocardial infarction. The intracellular domain of the notch receptor, NICD, can inhibit the signal transmission along with TGF-beta 1 SMAD3 pathway due to competition for the alpha SMA promoter. Let us see, this is TGF beta, and when this MAD3 is activated, then normally it brings in alpha SMA. The gene will be turned on, which is expressed. But if the same cell has got NICD, no signaling is turned on, it allow alpha SMA expression, but it do not allow SMAD3 to bind on to it. So in this process, it will alleviate the action of TGF beta signaling on the production of alpha SMA. So the gene is same, expression is same, but based on whether TGF beta signaling is turned on or delta notch signaling is turned on, you end up getting alpha SMA expression, gene expression, but it can compete out. Rather, you can say the NICD has got a higher affinity for the alpha SMA promoter than pSMAD or the SMAD3. So it can compete out and also trigger a more robust regenerative response. So differences between normal and hypertrophied CMCs. We know when there is an injury that takes place, fibrosis can happen in heart, and which will be cleared in the case of regenerating zebrafish. But in the case of mammal, the fibrosis stays, and that can debilitate the contraction of the heart muscle. Now, even in animals which do not regenerate, even though there is fibrosis, the existing cardiomyocytes, they have the potential to undergo something called hypertrophy. That means just like, you know how a balloon look like. You take a balloon, it is very flaccid, but if you blow air, it will become bigger and bigger and bigger. So we can call it as a hypertrophy. The balloon got hypertrophied, but the number of balloon do not change. Number of balloon remains 1. But earlier the balloon was, you can happily put in your pocket, but once you blow the balloon up, you cannot put in your pocket. Means it can occupy more space. So this is the principle of hypertrophy. But the muscle remains muscle, but now it can cater a larger area. That is the principle of hypertrophy. So normal cardiomyocytes, it responds to the action potential. Soon after a potassium channels that are opened, there will be an action potential. Whereas in hypertrophied cardiomyocytes, the action potential is occurring, but it do not come to the resting stage very fast. It will stay for a longer time. So the hypertrophied cardiomyocyte cells differ in normal CMCs in terms of electrochemical features, and they have a longer period of vulnerability to extra contractions. That means it contracted. If it came down immediately, that is called repolarization. This is depolarization and then repolarization. If it did not come back to the normalcy, it will not contract again. They have a refractory period before which it can respond to the injury. In a simple sense, if I say I gave you breakfast at 9 o'clock and you did not finish it until 1 o'clock, 10, 11, 12, 1, 4 hours you are having breakfast. Now how will you have lunch at 12 o'clock? You will not be able to. So you have to have a gap, and that should digest, and then you can finish. You can go for a second round of food. Same logic applies. Hypertrophied will not respond to their relaxation takes longer time, longer period of vulnerability to extra contractions. This is due to change in the amount and conductive properties of the potential dependent potassium channels. Because of this hypertrophy, the potassium channel distribution can get affected. Hence, they will not come to the refractory period very fast. Means they will have to wait for a longer time. And if you look notch signaling in angiogenesis, we all know angiogenesis is a necessary step in every regeneration, because angiogenesis is nothing but formation of new blood vessels, for which the endothelium comes into picture. Endothelial cells have to proliferate, then assemble into blood vessels. This is how the blood vessels are formed. VEGF is an important angiogenic factor, vascular epithelial growth factor. VEGF triggers the expression of DLL4. DLL4 is what? The ligand for delta notch signaling. DLL4 ligands in the leading tip of the cell triggering the notch in the adjacent stack cells. So when you have this ligand, that is VEGF, present, and it will trigger the expression of another ligand DLL4 in the tip of the cell, and that can influence, that can tell the target cell or the juxtacrine or adjacent cell which is having a notch to have a notch signaling, adjacent stack cells. So the direction of migration of the tip cells is determined by the CXCR4 chemokine receptor. Chemokines, we know, chemokines attract the cells. When chemokines are released, it's almost like a bait, how you do fishing. So you have a rod and a hook and a bait, you have put it. So but ultimately, if a fish is in your hook, it will come to you. So you can tell that, you know, the gradient, wherever the hook goes, doesn't matter. With the fish is there, you can bring the fish back to you. Same logic applies. The chemokines bring a cell where it want the cell to come. It will attract. Just like you put a light, the insects come into the light at night, right? Something logic. Same logic. So you should understand there are veins and there are arteries. Both require, you know, angiogenesis. Angiogenesis means not just vein, not just artery. Both are needed when it comes to angiogenesis. So in these cells, when they are growing, actually this blood vessel is growing, okay? When it is growing, the tip cells, they have, and VEGF is present, and when VEGF is acting, VEGF has got its own receptor, they produce based on the CXCR4, which is a chemokine that allow the migration of the cells to a given direction. But these stalk cells tips, sharp edges or the pointed edges, they have got the DLL4 expression which bind onto that cells, which the neighboring cell that has got the notch signaling. It can trigger a notch signaling event, and hence they will continue to proliferate. So notch signaling cell will produce genes that will allow the progenitors to proliferate. Hence you will have more of endothelial cells and more of the blood vessels that will be triggered, and it is decided by VEGF. The DLL4 expression is decided by VEGF availability, and DLL4 in turn allow the notch signaling to kickstart. So this is how the migration of the blood vessels or the growth of the blood vessels takes place. So if you derive the conclusion, the notch signaling pathway mediates cardioprotective effects at various stages of myocardial infarction. Ischemic damage to the cardiomyocytes at the reactive stage and also during scarring stage. In the early stages of myocardial infarction, notch causes a decrease in the oxidative stress by regulating the number of mitochondria and activating the MFN protein. Notch also activates the AKT pathway, which in turn is involved in producing antioxidant enzymes in the mitochondria. So it has to safeguard the cell. You don't want the damage to go further after a myocardial infarction. Finally, notch signaling is involved in the activation of a pathway associated with a AMP activated protein kinase. This also a role played by the notch signaling, which promotes energy reservation. That means cells are starving now because of the ischemia, and it should learn to conserve the existing energy. So the prevention of apoptosis is achieved by stimulating the production of survival factors such as PI3 kinase, phosphatidylinositol 3 kinase, and AKT pathway, and anti-apoptotic proteins. So the notch stimulates the survival of the cardiomyocyte cells, thereby reducing the damage because of the myocardial infarction. Otherwise the whole heart can suffer, but it will be localized. Notch 1 is able to reduce myocardial fibrosis by inhibiting the transmission of TGF-beta1 SMAD3 signals, reducing the production of ECM in the myofibroblast. We will study more about the regeneration in the next class. Thank you.